Welcome back, fellow scientists. So let's review our learning objectives before we get into our next lesson. So number one, history of DNA, done. 2, structure of DNA, done. 3, DNA replication, done. That was all chapter 12. Learning objective number 4, central dogma of biology, DNA to RNA to proteins to U, transcription translation. That was chapter 13. That's done. So now we are on to our last learning objective, tools and uses of biotechnology. So go ahead and get a new notebook page. I'm going to scroll down to the appropriate place on the smartboard presentation. So first thing we have to do, define biotechnology, obviously. So I'm going to define it as manipulating DNA to benefit humans, right? There are other definitions out there. Biotechnology is much more inclusive, but that's what we're going to talk about for this course, is mainly focus on the DNA aspect of biotechnology. And of course, we always want to benefit humans because we're the most important things in God's creation. Beautiful. So that's biotechnology. So this video, we're going to look at one tool and one process. So our tool that we're going to look at with this video are restriction enzymes. So go ahead and write down the fact that restriction enzymes cut DNA at specific sequences. So I'm going to give you 4 examples of restriction enzymes, and you'll be able to very easily, with these examples, see what they do. So first 2 restriction enzymes that we're going to talk about are NORO1 and SMA1. Those are the actual names. Just write them down. I'll explain how we get the names here in a second. But what they do is they are going to clamp onto DNA, and they're going to spin down the double helix, and they're going to look for this specific sequence, right? Restriction enzymes cut DNA at specific sequences. So NORO1, that restriction enzyme, that protein, that little Pac-Man, if we like to think about it that way, is going to clamp and is going to spin and is going to look for that sequence, ATTA. And what it's going to do when it finds that sequence, it's just going to, it's going to sever both sugar phosphate backbones right there. Okay, SMA1, same thing, except for it's going to look for a different sequence. It clamps on, it spins down, and it looks for CCCGGG, right? And when it gets there, it's going to cut. And both of these enzymes are going to produce what are known as blunt ends. Okay, the opposite of blunt ends in biotechnology are sticky ends. We'll get to that in a second. But you can see it's blunt. It's like cut off. There's no stickiness. There's no sharpness. It's just like it's a sheer cliff. If you're a little guy walking up here, right? Blunt ends. Okay, so that's NORO1 and SMA1. Next 2 enzymes that we're going to talk about are EcoR1 and HIND3. Now, with EcoR1, I'm gonna camp out on this a little while because this is how we're going to figure out how these restriction enzymes are named. So restriction enzymes are naturally occurring. They're naturally occurring in bacteria. Bacteria just make these normally. Restriction enzymes are nothing that humans made just out of nothing or by themselves or in their own mind. They discovered that bacteria naturally make these, and they were like, huh, well, that's kind of cool. We can use these to benefit us. So let's do that. So naturally occurring in bacteria. Well, question number 1, why would they naturally occur in bacteria? Well, if you'll remember all the way back to discovery of DNA and the Hershey-Chase experiment where we talked about the virus inserting the transforming factor, which we now know is DNA, into the bacteria, and the bacteria, like gets taken over by the viral DNA, and the viral DNA makes the bacteria make new virus parts, and then in the end the bacteria explodes. We call it lyse, but we can say explode because it's much more dramatic. So if you're a bacteria, you don't want to explode. That's a bad thing. Okay, so how do you stop from exploding? Well, you cut the viral DNA. So restriction enzymes are kind of like the immune system of a bacteria. The virus injects the DNA, and then the restriction enzymes from the bacteria, they cut, and then all of a sudden the viral DNA doesn't work. Okay, so that's thing number 1. Thing number 2 then is how we name them. So we name them after the bacteria where we found them. So EcoR1 is found in the most famous bacteria ever, E. coli, right? And it was the first restriction enzyme found. So HIND3, different bacteria, SMA1, different bacteria, NORO1, different bacteria. So anyways, EcoR1 and HIND3. So let's go ahead and let's look at the sequences that they find. So EcoR1 is going to clamp and is going to run down the double helix, the twisted ladder, the spiral staircase, and it's going to look for GAATTC. But it's not going to just do a straight cut. It's not going to cut through both sugar phosphate backbones at the same point. It has a little bit more finesse. So it's going to cut through this sugar phosphate backbone, and then ever so delicately, it's going to sever these hydrogen bonds between these bases right here, and then it's going to cut through this sugar phosphate backbone. Hind 3 is going to do the same thing, except for it's going to look for this sequence. It's going to cut, it's going to cut, it's going to cut, until we get something that looks like this. I drew in the one for EcoR1, you can draw in the one for Hind 3. So these are called sticky ends. Now, these bases, they want to pair with other bases. They're kind of like magnets. They're attracted to the bases that they should be attracted to. So that's why they're called sticky ends, because if they get anywhere close to each other again, they'll just go right back together. Okay, so those are restriction enzymes. Those are the classic restriction enzymes. Those are the ones that were discovered first in like the 70s and 80s, 1970, 1980, stuff like that. But you may have heard of a newer restriction enzyme that has been kind of in the news and making headlines and editing human embryos and different things like that. So CRISPR-Cas9 is also a type of restriction enzyme. Now here's what makes CRISPR-Cas9 so cool. It used to be, before we discovered CRISPR-Cas9, that we were locked into these restriction sites. So if we were studying a gene and there wasn't a restriction site close to that gene, for a restriction enzyme that bacteria naturally make, then we were kind of stuck. We were up a creek without a paddle. There's nothing that we can do. Because we couldn't program these restriction enzymes to cut wherever we wanted to. They were locked in. They're going to look for that sequence, and they're only going to cut that sequence. Aha! Along comes CRISPR-Cas9, which is actually a programmable restriction enzyme. Now again, this is naturally occurring in bacteria, and we just recently found it, like 2015, 2017, somewhere in there. Dates are kind of fuzzy, but that's okay. But the cool thing about it, the main thing that it does, is it has this guide RNA that's associated with the protein. So guess what? If we change this guide RNA, if we make these enzymes with different guide RNAs, then we can cut absolutely anywhere that we want to. And so we're not limited anymore by the restriction sites that bacteria naturally make. Now, because we have identified, because we've isolated, because we know about CRISPR-Cas9, now we can program a restriction enzyme to cut absolutely anywhere that we want. So that's kind of cool. All right, so that's our first tool, is restriction enzymes. So now we're going to go on to our first process, which is transformation. So transformation is making a cell produce something that it normally does not. Obviously, by that something, I mean protein, right? Or a protein that performs a certain function, which we'll see in our second example. So transformation, making a cell produce something that it normally does not. So here's one practical example. So let's say that you have diabetes, and let's say this is before biotechnology. This is before, like, the 70s and 80s and stuff like that. How did you get insulin? Because people who have diabetes, their cells don't uptake the sugar from the bloodstream like they should, and so they need insulin, which is normally made by their pancreas, but for some reason their pancreas isn't making insulin, so they need to take it, inject it into their bloodstream to make their cells intake the sugar. Okay, so what they used to do is they used to go to slaughterhouses, sheep and cow slaughterhouses specifically, and they would collect the pancreases from the discarded carcasses of the animals, and they would harvest the insulin from that, they would purify it, and they would put it in a tiny little medicine bottle called a vial. And that's how they got insulin. Now you had to process like pounds and pounds and like tens of pounds and hundreds of pounds of pancreases to get a usable dose of insulin. So that's just not viable, right? So along comes biotechnology, and they say, oh, well, if we could make something else make insulin for us, that would be amazing. So that's what they did. This was actually the first commercially produced biotechnology biotechnological product. So what they did is they took human DNA and they found the human insulin gene, and they cut, right, the insulin gene out of human DNA. And then they took bacterial DNA, which you'll remember is in a circle, and they used, this is key, they used the same restriction enzyme to cut. And of course, these are restriction enzymes that make sticky ends. Okay, and so then they just kind of mix the human DNA, which has the insulin gene in it, and the bacterial DNA, which used to be in a circle. They just kind of mix those. And then sometimes you would get something that looks like this. Sometimes the sticky end of the human insulin gene would find one sticky end of the bacterial DNA, and then the other sticky end of the human insulin gene would find the other end, and it would be amazing. So then what they did is they took this plasmid, and let's kind of shrink it down to size a little bit. And they just kind of incubated it with the bacterial cell, and they did like scientific magic. I mean, it's not really magic; it's actually science, but I won't go into depth. And they did some scientific magic, and they made the bacterial cell take in the plasmid with the human insulin gene. And so now, all of a sudden, we have a bacteria that has a human gene in it. We have a bacteria that's making insulin. So now we just grow petri dishes and we grow vats and vats of these bacteria because the bacteria just automatically reproduce through binary fission. Right? And so now these bacterial cells are pumping out insulin, and then all we have to do is we just have to harvest the insulin from the vat of bacteria. And so now we don't have to go to slaughterhouses. We have an almost endless supply of insulin at a relatively inexpensive price. So that's number one. And Mr. Leo, I am not excited. I'm not convinced. I don't have diabetes. I'm not planning on getting diabetes. I'm going to exercise. I'm going to eat right. Blah, 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 blah. Phooey on that. Okay, fine, whatever, you uncompassionate person, you. What about this? Okay, so there is a jellyfish that normally makes a protein that glows green under certain conditions, specifically ultraviolet light. So it's called a green fluorescent protein. Oh, that's kind of cool. Okay, so what if I told you that we could take that gene and we could put it into these bacteria? This is a petri dish full of bacteria, genetically modified bacteria. And when you shine an ultraviolet light on it, all of a sudden the bacterial colonies glow green. Well, Mr. Leo, it's bacteria. I mean, who really cares? What if I told you that we actually do this lab? We make glowing green bacteria in AP biology and in biotechnology. Okay, well, that's kind of cool. You know, it's still bacteria. Okay, fine. What if I told you that you could go to a pet store and you could buy glow-in-the-dark fish that have this gene, this green fluorescent protein gene, and there are obviously other things in nature that glow different colors. So we can have glow-in-the-dark fish. You have a little aquarium light, you have the ultraviolet light, you know, as the light source, and all of a sudden you have glow-in-the-dark fish. Okay, well, that's kind of cool. What if I told you that we can make glow-in-the-dark mice? Okay, that's sweet. I mean, not if you're the mouse. It's sweet if you're an owl looking for a mouse. It makes it pretty easy, right? Glow-in-the-dark rabbits. Okay, that's pretty sweet. Glow-in-the-dark. Now we're into primates. Glow-in-the-dark chimpanzees. Mr. Leo, could we make a glow-in-the-dark human? Technically, probably. Ethically, no. Because you have to do it when there are one-celled embryos so that every single cell has a green fluorescent protein, and you can't get permission from a one-celled embryo. And I mean, that child would be totally disadvantaged playing hide and seek, and it's just, it wouldn't be right, wouldn't be right. But one kind of cool scientific application of this is that we can attach that green fluorescent protein gene to another gene. So if we're studying like the development of an embryo and we want to see when certain genes are active and when they're not active, we can genetically modify that embryo. We can genetically modify that gene. Obviously, these are not human embryos. It's unethical to experiment on human embryos. So these are like mouse embryos or something like that. Those are fruit fly embryos. Well, those aren't fruit fly embryos because that's a vertebrate. But anyways, mouse embryos, we'll go with that. So we can attach that gene, that green fluorescent protein gene, to another gene to see when it's active. So we can see that whatever gene the scientists are studying is active right here, and it's not active all throughout here. So I don't know if you guys have ever heard of Science with Tom, but it's amazing. I show his videos in my class, mainly in AP biology. But the next video on the playlist, you don't have to watch, but it's a wrap all about plasmids. By Science with Tom, and it talks about how we can insert this green fluorescent protein gene and into and associate with another gene, and then have that glow as we need to, and we can use that to study. So, anyways, watch that if you want to. It's pretty short. It's like 2 minutes. It's really well done. It's very, very interesting. So, thank you so much for watching. This has been Mr. Leo, and I will see you on the flip side.